I'd like to start by thanking the organisers for inviting me to speak at the conference. I'm going to talk to you today about different strategies and ways in which we thought about annotating and interpreting non-coding variants in rare disease. Our current approach to clinical genetic testing is almost exclusively focused on regions of the genome that directly encode proteins. Now, I'm not going to pretend this strategy doesn't make perfect sense. It's a very small amount of the genome to assay, around 1.5%, and within this region, variants likely have a very large effect. They directly impact the protein coding sequence. And crucially, we have this beautiful amino acid code that means that our effect is one that we can predict. We know for any single nucleotide change, for example, whether that's going to be a silent change, lead to a change in amino acid, or create an early stop codon. Even when we have whole genome data, we currently focus only on the protein coding regions. Here I'm just showing you the tiering pipeline to identify likely diagnoses from the Genomics England 100,000 Genomes project, where to actually get into one of the top 3 tiers, a variant has to be protein altering in at least one transcript. So why are we so protein coding centric? The main reason for that is because interpreting variants in non-coding regions is actually pretty difficult. So the non-coding genome is massive. So the genome is around 3 billion base pairs. So there's an awful lot of variants. And actually a lot of that genome has some kind of annotation. So over 3/4 of it is actually transcribed into RNA, and over 6% of it is exonic. And most of the variants we find in non-coding regions have very little or no effect, which means that genome-wide tests for rare variants have almost no power. And thinking about those advantages of our amino acid code in protein coding regions, we don't have that in the non-coding space, which means that for any individual variant, it's very hard to define the effect that it has and the gene on which that effect acts. But we know from the literature that there are countless examples of variants in non-coding regions that cause disease. And here is just a subset of papers that I found in a short literature search. However, it's worth noting that these examples that we see in the literature and also in databases such as ClinVar are highly biased and very limited. And there's 2 key ways in which they are biased. So one is that they're biased towards genetically heterogeneous disorders. So the majority of the non-coding variants that we found that cause disease have been in individuals where we know which gene is likely causing their disease. So for example, neurofibromatosis type 1, we know that over 90% of individuals have a coding variant in the NF1 gene. And where we haven't found a coding variant, we've actually gone and screened a larger region of the gene and found new diagnoses. For the remainder of the variants that we found in the non-coding regions, they're actually mostly very close to the coding sequence because they've been picked up in panel and exome sequencing within the buffer region surrounding the coding sequence. And actually, given these biases, we're incredibly far from knowing the full spectrum of mechanisms through which non-coding variants cause disease. And these biases in the databases also impact our ability to build non-coding prediction tools, so in silico tools that actually predict the pathogenicity of non-coding variants when they're trained on truth sets that are very small and very biased. So we have to take the output of these kind of non-coding prediction tools with a grain of salt. So how do we tackle the non-coding genome? How do we begin to find new diagnoses for patients in these regions? And I'm going to take, you through 4 different points on this theme. So first of all, rather than looking at that entire genome, so those entire 3 billion base pairs, we can actually focus on regions or elements that have clear, important, large regulatory roles, where variants are likely to have a large impact and where we know which gene they regulate. So we know that each gene is regulated by a whole host of different regulatory elements. So whether that's the untranslated regions shown here in green, the promoter region directly upstream of the 5' UTR, cis regulatory elements that are more distal, either enhancers or insulators, and then this is all packaged up into this regulatory domains or TADs, so topologically associated domains that control the regulation of each individual gene. But many of these regulatory elements are hard to link to the regulation of specific genes, particularly these TAD boundaries and the cis regulatory elements. They also may be context or tissue specific, and particularly in these cis regulatory elements, there's high levels of redundancy, and that is enhanced for the most dosage sensitive genes. So a lot of our work is focused on what we think is actually quite a tractable region to start with, which is the untranslated regions or UTRs. So these are the regions directly up and downstream of the coding sequence. They are transcribed into RNA, but not translated into protein. And they have incredibly important roles regulating the stability of the RNA, so how long it persists within the cell, the rate at which it's translated into protein, and also where it's localized within the cell, often leading to incorporation of proteins into different protein complexes. And these regulatory processes are mediated by different sequence and structural motifs and binding to external factors such as RNA binding proteins and microRNAs. But crucially, in terms of variant interpretation, we know which genes they regulate, they're literally part of them, and they also contain functional elements that RNA biologists have studied for decades that enable us to begin to predict variant effect. So our first look into variants in these regions was to focus on variants with a very specific hypothesized effect. And these were variants that create a start codon within the 5' ETR. So a start codon upstream of the canonical coding sequence start. And this was a paper that we released back in 2020 where we used genetic constraint to look at the negative selection acting on these variants that create these upstream start codons. And what we showed that these variants are under significantly stronger selection than all variants within 5' ETRs. We show that that selection is stronger in genes that are already intolerant to changes in dosage. So for these variants, we expect they disrupt ribosome scanning and lead to a decrease in translation. So this makes sense that we see them under stronger selection at upstream of these kind of loss of function intolerant genes. And we also show that variants that create a kind of distinct upstream open reading frames, this is where they have an in-frame stop codon with that start also within the 5' ETR, are under a less strong selection than variants that actually don't have an in-frame stop codon, and where a ribosome, if it started translating from the early stop codon, would still be translating across the coding sequence, either out of frame or in frame, causing an N-terminally extended protein product. So we then went on to find these variants in different cohorts of individuals with different rare disease. So on the left here, I'm showing you some variants that we found as part of that paper published in 2020 that we discovered in HDMD, first published by Gareth Evans and team from Manchester, where they found in a cohort of individuals with neurofibromatosis type 1, 4 different variants that were within the 5' ETR. And these are shown on this plot on the left-hand side here. And 2 of these variants, shown in red, created these early start codons. And 2 of them actually disrupted an existing one of these upstream open reading frames, which is a negative regulatory element. But all 4 of these resulted in one of these overlapping out-of-frame ORFs, so where there was not a stop codon within the 5' ETR, predicted to lead to loss of function and to then hence be the cause of neurofibromatosis in these patients. On the right-hand side is then a paper where we looked, this was a collaboration with the amazing Caroline Wright from Exeter, where we looked for these variants in the Deciphering Developmental Disorders, or DDD cohort. And these individuals, we found a very intriguing gene, MEF2C, where we found 2 different mechanisms by which these start-creating variants cause a complete loss of function and lead to developmental disorders. So that's showing how we can focus on specific regions or indeed regulatory elements to identify variants that cause rare disease in the non-coding genome. What we can also do is we can focus on individuals where we know which gene is likely causative. So I've already spoken about this in one sense. I spoke about the biases in existing databases, but we can also use this to our advantage. On the left-hand side here, I'm showing you PAC6. So PAC6 is the poster child of 5' ETR splicing. So here we can see that there is a buildup of different variants within PAC6 that are known to cause congenital aniridia. On the right-hand side is that example I just showed you of NF1 and neurofibromatosis type 1. And for both of these, this is where we have a single gene that we know that a patient is likely to have a variant in that gene, and that can help us to identify non-coding variants in those patients. So I'm going to take you through 3 different projects where we use both of these 2 ideas. So 1, focusing on clear, important regulatory elements, and 2, looking in these patients where we know 1 or a small number of genes that are likely to be the disease-causing gene. So throughout these 3 projects, we looked at the same different regions, and this is a schematic of those here. We looked at a 5' ETR and the introns of the 5' ETR, 3' ETR, and also its intron. We looked at an upstream promoter region, and then for 2 of the 3 projects, we also included the introns for the coding sequence as well. But what focusing on these regions allows us to do is also to predict the effect of candidate variants within each of these regions. So in the promoter region, we looked at loss of transcription factor binding using a tool called Fabian. In the 5' ETR, we looked at variants that disrupt translation, such as these ATG creating variants I've spoken about already, using a tool that we created called UTR annotator. In the intronic regions, we looked for variants that were disrupting splicing using Splice AI. And in the 3' UTR, we looked for variants that were disrupting RNA binding, disrupting microRNA binding, or changing polyadenylation. And also across all of these regions, we looked for variants that had particularly high CAD or Philo-P scores to identify things where we didn't predict a priori a likely mechanism, but they might pinpoint important bases. For all 3 projects, we also used the same data set. We used the Genomics England 100,000 Genomes project, which has 100,000 genomes, but from a mix of rare disease and cancer. So in total, there were around 20,000 rare disease patients in this cohort and around 17,500 cancer patients. So in the first project, this was a wonderful collaboration with Diana Barali and Jenny Law down in Southampton and Hilary Martinez at the Wellcome Sanger Institute in Cambridge. And in this project, we wanted to find what we call recessive second hits within this Genomics England project. So what we did here is we took undiagnosed trio probands from GEL, so ones that had been recruited as part of a trio, so they had unaffected parents and an unaffected child. And in this child, we looked for ones that had a single coding loss of function or ClinVar pathogenic variant in a gene that is a known recessive cause of a developmental disorder. And we found 2,705 of these examples. And in each of these individuals, in those specific genes, we then looked for a variant on the other allele in one of those non-coding elements that I described earlier. And what we did is we then used these region-specific annotations to filter down these variants to ones that we think are most likely causative. So we found 52 variants in total after this filtering approach, and we then performed a strict clinical review and found 5 specific variant gene probands variant gene pairs that are a probable cause of the patient's phenotype, and many more that were possible. So here I'm just flashing up those 5 likely diagnoses where we have the details of the gene, the phenotype, the coding variant, and also the non-coding variant that we found in trans. 4 of these were intronic to the coding sequence, and 1 was found within 200 base pairs just upstream of the transcription start site. And this was in the gene called GAA. And actually, enzymatic assays confirmed this was the likely causative gene in this patient. However, when we then looked into this further, there was actually a 5' UTR splice altering variant that was more likely to be the second hit than this core promoter variant that was in cis. And we also found these 2 variants in cis, and along with being in trans with a coding loss of function variant in 3 different patients within GEL. So a short conclusion for this project. So we created a systematic pipeline to identify these common, non-coding second hits. This finds new or misdiagnoses, but we conclude this is not likely to be a common mechanism that will diagnose a huge proportion of undiagnosed rare disease patients. So the second project, we took a little bit of a step back. We had a broader number of genes here, and we focused specifically on de novo variants, these variants that don't exist in unaffected parents, but are created solely within an affected child. So this was work from a wonderful Alex Martin-Geary, who's a postdoc in the team, and she started with these undiagnosed rare disease trio probands, looked specifically for de novo variants of high confidence, and then focused on these non-coding regulatory elements that I showed you before, specifically in genes that are tagged as a likely a priori diagnostic gene for that patient. And these were flagged as green in the Genomics England panel app. So green means high confidence disease genes for a particular disease panel that was applied to that patient. We then again employed this region-specific filtering approach to try and find some new diagnoses. So we started off with a set here of 8,000 probands, and in these we found around 300 of these had a de novo variant, and after our region-specific filtering we found 11 potentially causative variants in 11 probands. So here on the right I'm showing you a plot which shows you the region that these variants were found within, and also on the upset plot the annotations for which they were included. And within these 11 candidate variants, 4 of these we class as positive controls. These are variants that we know have been already identified as likely causative. 2 of these are PAX6 5' UTR splicing variants. As I said earlier, this is the poster child, so these were classified as likely pathogenic. There is an RPL26 splicing variant that had already been fed back as a likely diagnosis, and also a MEF2C variant that we'd found previously as part of that paper that I discussed earlier. So just to tell you a little bit about this variant, because I think it's quite a neat mechanism. This variant creates a new start codon in the fibrometeor. It's one of these upstream AEG creating variants, but this is in frame with the coding sequence and adds only a small amount of sequence to the start of the MEF2C protein. But the reason this is pathogenic is that the N-terminus of MEF2C actually binds directly into the DNA for it to function as a transcription factor. So in order to validate, and so our prediction was that adding a small amount of sequence to the N-terminus disrupts this DNA binding and disrupts its function. So in order to validate this, Laura Ramos-Hernandez, who's a talented PhD student in Madrid, performed a co-transfection assay where she cloned mutant and wild-type fibrometeor upstream of a MEF2C gene, and then co-transfected that with a luciferase reporter gene driven by MEF2C upstream binding sites. And here on the right, you can see we actually found two variants with the same effect, each found de novo in 3 unrelated patients across 3 different cohorts, and both of these variants significantly decrease transactivation by MEF2C compared to the wild type. So of these 11 candidates, we had these 4 positive controls, and so of the remaining 7, 5 of these were actually looked to be likely new diagnoses after clinical review. I'm just going to talk to you about a couple of these now. So the first of these was SETD5. This is another 5'TR splicing variant. It has a very high splice AI score of 0.97. This was found in a 16-year-old boy who had a good phenotypic match for SETD5. We actually got in touch with the clinicians and they did a methylation assay which proved that SETD5 was a likely diagnosis for this patient. This variant had been identified by GEL, called a VUS, and transcriptomic studies had been suggested before these methylation confirmation studies were performed. The second variant is an SLC2A1 variant. This is another one of those variants that creates an early start codon. In this case, it's out of frame with the coding sequence. If the ribosome starts translating this upstream overlapping ORF, it will not be available to translate the coding sequence, causing a large decrease in translation. This boy was enrolled in 2017 at 9 years old, has an incredibly strong phenotypic match with SLC2A1. Again, we've been in touch with a clinician who's confirmed that this is a likely diagnosis. And this is so this gene causes GLUT1 deficiency syndrome, and this is actually a disease that is treatable by a change in diet. And GEL had not identified a variant for this patient previously. But actually, this exact same variant had previously been identified and published exactly the same year that the patient was enrolled into GEL. But it wasn't actually uploaded into ClinVar until it was found in another patient by Invitae in August of 2021. So actually, this patient, in theory, if this literature had been searched and found, could have been diagnosed 6 years ago and 6 years earlier and treated through diet. This outlines the importance of data sharing and uploading our variants and their pathogenetic interpretation into databases such as ClinVar. So in conclusion for this second de novo project, so we found a new diagnosis in only 9 of around 8,000 individuals, so about 0.1%. So we can find diagnoses using strict filtering in these non-coding regions. However, again, this suggests that we're not likely to change the world here. We're not likely to diagnose a huge fraction of undiagnosed patients by looking in these near coding regions. Just want to mention that we didn't include the coding introns in this project here, where there's likely to be quite a few more diagnoses. So I just want to quickly mention an important aside here. I've spoken a lot about untranslated regions, or UTRs, and I just want to mention that these regions are exonic. So there's a lot of confusion in the field where many people believe that what an exon is a region that codes for protein. And that is not a case. Actually, an exon is any region that persists within the mature RNA after introns are spliced out. So UTRs, coding sequence, and also the exonic regions of non-coding RNAs are all also exonic. So when we refer to whole exome sequencing, that is a bit of a misnomer because it's very much not whole. And we don't actually expect to capture the majority of UTR variants in exome sequencing data. We do capture some. The project we did in DDD was based on exome sequencing, but the majority of the UTRs are not captured. Just to kind of put this in graphical form, this figure on the left-hand side here, what I'm showing you is the height of each of these bars is the total number of base pairs of these regions with the annotations across the x-axis in the human genome. And then the proportion of each bar that is coloured in is the proportion that is included in the exome sequencing panel used by the UK Biobank. So as you can see, this encompasses a large proportion of the coding sequence, but very little of any of the other exonic annotations. And so what we really are capturing here is coding exome, not the whole exome. Okay, so the third of these projects where we've identified a cohort of individuals where we can predict one gene that one or a small number of genes that we think might harbour a causative variant. So here, Johnny Talbot Martin, he's a very talented undergrad student that worked with us, and this was in collaboration with Serena Nick-Zainal from Cambridge. And what we did here is we looked for individuals within GEL who had a somatic mutational signature that was suggestive of them having a BRCA deficiency. And within these individuals, we looked for those that didn't have a coding variant that explained that mutation signature, so a coding variant within BRCA. And what Johnny actually found was 9 different individuals with predicted deleterious splice-altering variants. So many of these were canonical variants that had been identified previously, but 2 of these were actually deep intronic splicing variants. And this was a strong enrichment compared to looking at the full GEL cohort, so how many background BRCA splicing variants we would expect. He did also find one of these upstream start creating variants and one variant that was predicted to disrupt a transcription factor binding site, but both of these individuals had an alternative diagnosis through a coding variant, so weren't thought to be likely diagnostic. So that's the kind of 3 different ways in which we've been thinking about how we can get these first 2 ideas of how we can tackle the non-coding genome. But what we've also been doing is developing tools and resources to actually help us do this variant annotation and interpretation on a larger scale. So the first thing we've created is this tool called UTR Annotator, and this was created by a wonderful PhD student back in my old lab at Imperial, Xiaolei Zhang. She translated my horrible Perl code into a plugin for the ensemble variant effect predictor. And what this tool does is it predicts, so if at any variant with a 5'UTR annotation from VEP, it will annotate its effect, whether it creates a start codon within the 5'UTR, or whether it disrupts an existing upstream open reading frame, either by disrupting the start or stop codon, introducing an early stop codon, or shifting the frame of that ORF. And actually, when she ran it over ClinVar, it actually annotated around 1/3 of all likely pathogenic and pathogenic 5'UTR variants, suggesting that a large proportion of these act through disrupting translational regulation. But that's all well and good and actually very useful probably to this community, but for many bioinformatic tools are not the most useful thing for variant interpretation. So we actually then built, so this is by an incredibly talented PhD student, Elson D'Souza, and he built the annotations from UTR into a visualization tool which we call VUTR. So here I'm to kind of demonstrate this, I'm just going to show you a screenshot from the output from the MEF2C gene. And here we can see across the top here we have some various different gene level details, links to external resources, details about the 5'UTR. And underneath that we then have the architecture of the native 5'UTR. This is a reverse strand gene, so on the left hand side we have this big dark grey coding exon box, the coding sequence box, and then this has 2 different 5'UTR exons. It has 2 existing potential upstream open reading frames encoded within the sequence, but both of these have a weak start codon match to the Kozak consensus. They're likely very lowly translated. What's underneath this then are every single variant within Nomad and within ClinVar that have also been annotated with UTR Annotator. Any variant that has a predicted effect from UTR Annotator has an expanded out row which shows you the effect that it has on creating or disrupting UORFs. As we can see, the variants within Nomad all create these weak Kozak consensus UORFs that are far upstream with the coding sequence, whereas the variants that we find in patients in ClinVar are all create these ORFs that overlap with the coding sequence. So we can see clearly how these might be pathogenic, but the ones that within Nomad are likely not having a large impact. We also then realised whilst looking at these variants, so every time I kind of spoke to a clinician about a variant that we found, the 5-UTR, they would immediately come back and say, well, how do I interpret that? How do I fit this within the ACMG AMP guidelines for clinical variant interpretation? Now, these guidelines are primarily very heavily protein coding centric. They were designed for use on what we do clinically test, which is also a lot of the rules are specific to variants that have a missense or a loss of function effect. So what we did, we put out last year a set of recommendations that are intended to sit directly alongside the ACMG AMP guidelines and detail ways in which you can adapt the existing ACMG AMP framework to apply for variants in non-coding regions. So this was a bit of a challenging endeavour. So here we had to write guidelines that were specific enough to be useful, but also could account for the limitations in our knowledge of disease-causing non-coding mechanisms. So they would apply across all different variants that we didn't even know existed yet. But we got together a lot of awesome people in the room. So this was an effort co-led with the wonderful Jamie Ellingford, who's in Manchester. We got together a load of people that are involved in clinical variant interpretation, particularly with interest in the non-coding space, and also brought in a couple of consultant reviewers, so the amazing Heidi Ream and John Greeley, who works on epigenetic data in New York. And what we did is Jamie and I sat down and drafted an initial set of recommendations, thought very heavily through all of the different rules and how they might be adapted, and then took this document to the expert panel. We had a lot of discussions, incorporated a lot of feedback in an iterative fashion, and then when we were happy with the document that we had, we sent this for extensive testing. I'll show you the amazing testing team in a minute. And then we sought input from the ClinGen sequence variant interpretation group just to check that we were then going to be aligned with the upcoming guidance from ClinGen and the revamped version of ACMG AMP. And then we put this out for community feedback before finally getting it published. So I mentioned our amazing testing team. Here are their names. These guys painstakingly took 30 different variants across a range of different non-coding regions, used data and tools that they were completely unfamiliar with, but gave us some incredibly valuable feedback that we then built into the guidelines to make them usable to people in a day-to-day setting. We learned a lot through this effort. So one thing we put a lot of thought in is kind of which variants should we be interpreting clinically. It's clear that we cannot tell clinical scientists to suddenly interpret variants that exist in the entire genome. That's just not going to work. They're already very overwhelmed. So we define what we call interpretable regions. And these are regions that have a very clear link to the gene of interest. And that gene has to be a gene that is a known cause of a patient's phenotype. We found out that with our testing team, the epigenetic data and the in silico tools that are needed to interpret these variants are incredibly difficult to access. And we also spoke right at the start of this talk about how they might also be biased and not that good for variant interpretation. So we need better tools and we need better access to those tools to allow us to interpret non-coding variants routinely. There is a huge amount of uncertainty around hypermorphic variants, variants with only a partial loss of function effect, and whether they might cause disease. What is the threshold of disease in any particular gene? And how variants within the non-coding space might influence penetrance of coding variants or themselves be lower penetrance than variants found in coding regions. And because of all of these uncertainties, variants will need to be reinterpreted as our knowledge evolves, and also our guidelines will also need to evolve with that knowledge. So finally, my 4 points of how we tackle the non-coding genome is that we need to be screening for these variants regularly in patients, and we need to share that data openly. Only through having the massive amounts of data are we going to be able to find out more about different disease-causing mechanisms and create the tools to be able to interpret them routinely. So finally, in conclusion, interpreting variants in the non-coding genome is hard, but if we do this sensibly, we can find new diagnoses for patients, although this is likely to be a very modest proportion of undiagnosed patients that are explained through these non-coding region variants. But it's also a very fun area to work in, and the more variants we find, the more we learn about regulatory mechanisms. We need to start with regions and genes that are tractable, where we think that we are likely to find a non-coding cause in a particular patient or a particular gene. Otherwise, we just have far too much noise. We need better tools, better ways to access the data that we need to interpret these variants routinely. And although we now have guidelines to help interpret them, they need to be refined and adapted as we have new knowledge. And for all of this to evolve and for us to move further in this field, data sharing and collaboration is key. So with that, I just want to acknowledge my awesome team at the Big Data Institute in Oxford and all of my funders, and thank you very much for listening.